Good morning, everyone. Hope you're all doing well and safe and you're in good health. I just wanted to go over this video, hopefully is brief. It's a review of all the materials or most of the materials you should be familiar with for the assessment on, depending on what period you are, either Monday or Tuesday. All right. So up on my screen, I have the study guide. So the two documents that will help you the most, and you're welcome to have them open during the test are, well, this one's not a document, but hopefully you've done this, the vocabulary terms for the unit, and then the study guide, which, which will be due, I believe, next Tuesday. So hopefully you finish these because they will come in the questions here, the answers to these questions will come in very handy during the test. Okay, so this document is split by topic: meiosis, mitosis, which are two different types of cell division. Meiosis is a specific or specialized cell division for your sex cells, sperm and egg cells. They're produced using meiosis, and those are the only cells in your body, in the human body, that go through meiosis. And you can kind of um, remember that because sex cells only are haploid. Okay, haploid meaning half of the chromosomes. So instead of 23 pairs, there's only 23. Okay, so your body cells, your somatic cells, which go through mitosis, have 46 individual and 23 pairs of chromosomes. And those are considered diploid. All right, so that's the big difference between meiosis and mitosis. Um, and so then you have to go over terminology like gametes. Those are the sperm and egg cells. How are they formed? Through meiosis. What does haploid mean? It's half of the number of cells or number of chromosomes compared to a full set, which occurs in mitosis. Okay, how many cells are produced when one germ cell? So meiosis, gametes, sperm and egg cells do not divide. Okay, in the case of twins, the fertilized egg can divide, but in germ cells produce gametes. So you have spermatocytes and oocytes. Spermatocytes produce sperm cells, oocytes produce egg cells. And with spermatocytes, you get 4 haploid sperm cells per division per meiosis of a spermatocyte. Oocytes are special or different because even though you get 4 cells, only 1 egg cell is produced, and the other 3, the remaining 3, are polar bodies. They're kind of like the extra information, just not able to be fertilized. Okay, so that's the answer to that question. Homologous chromosomes, you should know what those are. We distinguish chromosomes by size. They're paired in your nucleus by size. And so homologous chromosomes are the same size chromosomes, same number of chromosomes. How many pairs of chromosomes do humans have? That should be pretty simple. Pairs, 23, right? Karyotyping is when someone takes your chromosomes and lays them out and basically arranges them by size. Number 1 is the largest, the longest, and number 22 are the smallest pair. And then pair number 23 is your sex chromosomes. And if you're a woman, you have 2 X's. The X's are the bigger ones than the Y. So if you're a man, you have X and a Y. And here is where we also talk about the different types of genetic disorders that we've gone over, right? Klinefelter's. In karyotyping, you can also tell if the patient or the subject has any kind of genetic disorder, usually additional chromosomes or chromosomes that have been omitted or deleted or are not present. Okay, pedigree charts. Please know what these different symbols mean. One of the most important things that you should remember are carriers versus affected. So if a question asks about something, a disease or a condition that has been kind of inherited, and then asks who the carriers are half shaded versus affective who are fully shaded, right? And then so X-linked traits are genes that are carried on the X chromosome, the chromosome pair number 23, where the XX or the XY are the two possibilities for male or female. Okay, so if you have an X-linked trait and you're a woman, usually it means you're a carrier because you have two X's, so you're able to your healthy X is usually able to mask over the diseased X if we're talking about a disorder. And if you're a male, and this is the case in color blindness, and you have that X from mom that has the colorblind gene on it, or colorblind allele of the colorblindness gene, then you will almost always be colorblind. All right. So in colorblindness, women are usually carriers, and men in families where colorblindness is prevalent have it. Okay. What are carriers of traits? Those are people who carry the trait but do not show it. And how are they signified in a pedigree chart? We just went over that. All right. Karyotyping, easiest way to tell chromosomes apart is by size. Centromeres is the center of the chromosome. And again, what is a karyotype? We already went over this. Next topic, CRISPR. We talked about this during our meetings yesterday. I wish we had been able to cover this while we were still in class because this is one of the most interesting parts of science at the moment. CRISPR is basically how genetic manipulation is going on right now in labs. If we know where a gene is, and if we know the code for that gene, and if that gene is something that is causing disease or any kind of difficulty in a human, we can use CRISPR to go in and do genetic therapy. So CRISPR comes from, what is CRISPR? It's a protein machinery, basically, for the lack of a better term, that will go in and well, it's derived from bacteria, and bacteria usually have to repair their own DNA because they frequently get infected with viruses, and in the unicellular universe, a virus will come into a bacteria, infect the bacteria, and then inject its viral DNA into the bacterial DNA. And so the bacteria uses CRISPR to basically go in, detect the viral DNA, and then repair it using CRISPR. So what scientists have done is, actually, let me pause this and bring over a picture so I can explain it better. All right, so this is basically a depiction of CRISPR. The pink blob is a machine, which is a bunch of proteins that are specialized, that are working. So CRISPR will basically go over these go over the bacterial DNA, look for viral DNA, and then clip it and repair the bacterial DNA. So what scientists have done is take CRISPR, load it, tell it to, instead of looking for viral DNA, look for genes or codes that we want to get replaced, and then we program it to replace it with a certain other code. Okay, so in a future that's not too far away, actually right now people are using this to look for cancer genes and replace them with healthy genes in people with cancer. They're doing that. They've, as we discussed in our class, a Chinese scientist actually change the DNA of twin sisters to make them immune to HIV. And then later on, when this becomes normal, you could probably go in and change features of yourself that you want to improve. Okay, so more reason to do well in school and go make that money so you could do things like that. All right. Types of genetic dominance. These are easy questions that you can score a lot of points on. Complete dominance is recessive, is always dominated by the dominant allele of a gene. So alleles are again a flavor of gene. So the gene for eye color has a blue eye color allele versus a brown eye color allele. Okay, so complete dominance is when the recessive is completely dominated by the dominant allele. Incomplete dominance, this is when you get a mixture. Okay, this is where red colored flower cross pollinates with a white colored flower. The red color is the dominant gene or allele of the color gene, but it's incomplete dominance, so you get a little bit of the white flower's genetic info, and therefore you get a pink flower. Okay, codominance is when both alleles are dominant. In our presentation, it was the white chicken and the black chicken producing the chicken with the uh, speckled uh, feather pattern. So both white and black feathers, right? Why is skin color a polygenic trait? Skin color, polygenic, first of all, means many genes. So skin color is not just, there's not one gene that in your DNA that codes for skin color, but your skin color is a product of many genes working together. And this is basically because skin color is an adaptive mechanism depending on where you come from, where your DNA has been historically. So if your DNA migrated to where you were born through the northern hemisphere, where there's little to no sun, uh, your your skin adapted to have little to little pigment because you needed to absorb as much sun as possible. If your DNA traveled through, let's say, this close to the equator where there's plenty of sun, your skin adapted to produce pigment to protect itself from the harmful rays or harmful effects of the sun, but also be able to absorb sun because you need it. You need sun energy to create vitamin D. All right, lactase, the last topic. Lactase. So lactase is an enzyme. Lactose is the sugar. Lactase breaks down lactose. So an enzyme is basically a specialized protein, and your body has to code for lactase in order for you to have lactase to break down lactose. Okay, lactose intolerance, therefore, in humans means that your body is not making the enzyme lactase. Okay, and so a lot of people become lactose intolerant later on in life, and that's that's something that we were on the cusp of going over before we left. And basically, your body stops making lactase to break down lactose. Um, based on many different things, interesting things. The more interesting part is some humans stay, their bodies continue making lactase, and we say those are lactase persistent individuals because regardless of age, they can digest lactose because their body keeps making that gene. Okay, and which parts of the world's population? This was in the Click and Learn. And I'm sure you can guess, the herders, the people who keep cattle for dairy purposes have a prevalence of lactase persistent individuals. Okay, and I encourage you to look over that Click and Learn and learn a little bit more. It's very interesting. It may, be personal for some of you because maybe your family comes from those regions of the world. All right, so this is in a nutshell a review of the test or of the material that will go into making the test. And the test will be multiple choice and some free response. It won't be difficult. It'll be um, um, monitored by me, but it'll be online. So it'll be a lot of it will have to be on the honor system, right? So please hang on or look out on Google Classroom for more instruction about the test um, before the test is given on Monday, all right? If you have any questions, like always, please email or comment in Google Classroom and I'll get back to you as soon as possible. Have a good day. Have a nice weekend.